Our next speaker is Dr. Michael Kahana, who is the Edmund and Louise Kahn Term Professor of Psychology at the University of Pennsylvania and the CEO of NIA Therapeutics. His work has reshaped how we understand memory, and he'll tell us about using AI-guided closed-loop stimulation to enhance memory in patients with brain injury. Let's welcome Michael Kahana.
Well, it's wonderful to be here. I don't think I lived in Boston for 10 years. I came here in 1993 to do a postdoc at Harvard, but I don't think I've ever been to the Harvard Club. So it's wonderful to be in this auspicious place. I'm now in Boston more regularly because I'm on a leave of absence from the University of Pennsylvania to, there we are, to serve as CEO at NIA Therapeutics. It's also wonderful to be here with friends from Brandeis University and the University of Pennsylvania. After I came here for my postdoc, I joined the faculty at Brandeis, where I had the pleasure of being a colleague of Margie Lackmans and Art Wingfield and Bob Sekuler, and I came back last Friday for Bob Sekuler's Festschrift, which was wonderful. So I was actually here just a few days ago. After being at Brandeis for 10 years, I went to the University of Pennsylvania, and that's where I've been on the faculty until I took the leave that I'm on now to try and advance a therapy for memory loss that grew out of research that was done in my lab. So by way of a brief disclosure, I have equity in NIA Therapeutics, so that's something you should be aware of. I want to start by sharing with you my journey into neuroscience, because actually I've known Marty since before I had any real interest in neuroscience and the brain. We were both growing up in the field of human psychology of memory, and I studied at the University of Toronto with Ben Murdoch and Endel Tulving, memory. And then when I came to Harvard, I worked with Bill Estes, and there was no brain science there, except before I embarked on that journey, I had the good fortune of taking Jeff Hinton's neural networks course, and that introduced me to some of the ideas in AI that later penetrated into my work. But my real journey into neuroscience began here in Boston when I was at Brandeis, and I met a young neurosurgeon by the name of Joe Madsen, who I believe just retired at Children's Hospital. Brilliant, charismatic neurosurgeon who invited me to speak at Harvard Medical School about memory, the science of memory. And I described mathematical models of recognition memory. I don't think anybody was interested except for Joe. But then he took me to the ninth floor, the north wing of the building, and introduced me to what he does, which is implant electrodes in patients, young people who have intractable epilepsy. And that began a collaboration and a journey that preoccupied me for the better part of 25 to 30 years. In this work, patients with epilepsy get implanted with intracranial electrodes, either intraparenchymal or subdural electrodes going on the surface of the brain or penetrating the brain. And they're monitored for seizures for a period of anywhere from 1 week to 3 weeks. And I realized I could come in and do memory experiments on these patients and learn a lot about the neural basis of memory. And to Marty's point, the interest in studying memory in this way is to really be able to measure at a very fast time scale the dynamics of activity related to variation in performance. And the main theme of my work really resonates with Marty's presentation, which is that you may worry about whether your memory is better or worse than somebody else's memory, but in fact, there's tremendous variability in each of our memories from moment to moment, from day to day, week to week. I won't tell you about a 25-year journey of studying the electrophysiology of memory, but I'll say that in 2014, an opportunity arose to expand the scale of human neural recordings in the study of memory dramatically because the Brain Initiative and the Department of Defense under DARPA had a call for applications for very large grants. I was PI of one of these large grants called the Restoring Active Memory Project. And in a period of 4 years, we studied memory in 422 neurosurgical patients at 9 different hospitals. We had collaborations with 11 different institutions, collected data from 2,400 experimental sessions, over 1,000 brain stimulation experiments. And I will say one of the things that I'm proudest about from this study is not just the scientific discoveries, about 35 to 40 papers from my group, but the fact that we decided at the very start in 2014 that we would publicly share all the data and all the code, and we have done so. And there have been dozens and dozens of papers reanalyzing. This is the largest human memory intracranial data set in the world, and it's all available on openneuro.org. And I think it's a testament that my trainees have benefited rather than being harmed by data sharing, that I encourage everybody to do that with their data. These patients have electrodes all over the brain. These are these green dots on the spinning brains is just an illustration of one experimental data set from about 100 patients. In total, at this point, we've collected data from, as I mentioned, 422 on the DARPA RAM project, but continued work in my lab, about 700-800 patients. As Marty noted, the key question that emerged from a lot of the research on memory is what I call the variability puzzle, which is, why is it that your memory can be so variable from moment to moment, from day to day? And I did a lot of work on this in the psychological laboratory at Penn, where I studied this problem, and many papers written about it. But the basic conclusion of that work is that you cannot actually account for most of that variability using known experimental factors, or factors such as whether somebody got a good night's sleep or whether they're anxious. Yes, those variables are important, but there's underlying variability due to the network physiology of the brain. There's variability that cannot be accounted for except based on autocorrelated activity in the human brain, or autocorrelated activity that you measure in a behavioral task. And one way to study that is you give people a list of items. Marty described some tasks that are mostly focused on reaction time measurements. I'm focusing here on accuracy measurements in a free recall experiment. You give people a list of items to study, you distract them for a period of time so you're not measuring short-term or working memory, and then you ask them to recall the items in any order they like. That's the free recall task. Many neuropsychological measures are variants of the free recall task. It's very sensitive. And you can record from neural activity in the brain across many electrodes and at many frequencies of neural signal. And from those, you can create these essentially brain maps of what the brain physiology looks like for items that people remembered and what it looked like for items that they didn't remember later. And you can ask, can we use machine learning, standard machine learning methods, to forecast whether memory will be, whether an item will be remembered or an item will be forgotten? And it turns out that you can do that. Our first paper showing this was published back in 2017. We've done many, many studies on this. Here's just an illustration of one sample data set from one subject, where the y-axis is the probability that the model thinks the item is going to be remembered. And the red dots are remembered items, the blue dots are forgotten items, and the x-axis is indicating the first 8 items on the first 8 lists. And you can then compute an ROC curve, which shows that the model is able to differentiate between items that were learned and items that were not, that were recalled, and items that were not recalled in this subject and in another subject. And you can do this in hundreds of subjects. It turns out that this kind of decoding is reliable in every subject that we've investigated, basically. If you take, I think there's something wrong with this slide, but that's okay. If you look at the curves in the middle of this graph that say upper, middle, and lower, those are so-called serial position curves. They show the primacy effect. The early items are remembered better than the middle items. They don't show a recency effect because we distracted subjects. And the three curves show the difference between when the forecast model said that your memory was going to be in its top third, middle third, or bottom third. And what you see is a dramatic difference in performance between the top third, the middle third, and the bottom third. Marty was referring to 2 to 5 years of the variability is 2 to 5 years of degenerative changes in function across degeneration. Our estimates are similar to Marty's. I would have said closer to the 5-year range in terms of how much of a difference you get between trials that are forecasted to be good versus bad. And it turns out you can do the same thing at both encoding and at retrieval, and at the classifiers, the models that predict encoding also predict retrieval. The models that predict retrieval also predict encoding. So there's a common core of neural activity in the brain that fluctuates between good and poor memory states to explain both variable encoding and variable retrieval. We learned a lot of lessons from the DARPA project, the Restoring Active Memory project. We did a lot of studies using electrical stimulation of the brain, and I just summarize a few of the lessons here to be brief. When we decided to first experiment with electrical stimulation, our target was the hippocampus, entorhinal cortex, parahippocampal gyrus, perirhinal cortex, all of those MTL regions. And what we found is that stimulating in those areas almost always impaired memory. That was true for both spatial memory and verbal memory. It was true for both verbal memory with unrelated items and semantically related items. In general, we showed that we could impair memory, not very useful in this for the purposes of this conference, that you can impair memory. There could be uses for impairing memory, I suppose, but we won't talk about those today. It turned out that lateral temporal cortex was a target that when you stimulated there, sometimes it had no effect, but when it did have an effect, it often boosted verbal memory, verbal episodic memory performance. But the main discovery that we made from these studies was that when stimulation of the brain was delivered during good moments, not surprisingly, memory got worse. But when stimulation was delivered during poor moments, sometimes it didn't do anything. Sometimes it made memory better. And when you control for, you have to do a bunch of controls for regression to the mean, that effect persisted. So we then decided that we would actually create a closed loop methodology to rigorously evaluate this. And that's what we did. We used the same machine learning decoders to select the stimulation parameters. That's a whole other story. And then we delivered stimulation in closed loop based on the machine learning model. That was back in 2018. We used an externalized device that's shown here. Took us a lot of time to develop it. And I won't go into all the details because we'll run out of time. But basically, the methodology was you look at the state of first you build a model. You do record-only sessions. You build a machine learning model that forecasts good and bad memory. Then in the closed loop session, you record data from the brain. You make a forecast. Is memory going to be good or bad? If memory is predicted to be poor, you stimulate using parameters that were selected to optimize memory as best as you could. And when you do that, we found that when you stimulate in lateral temporal cortex, you make memory reliably better. When you stimulate in other areas, mostly in the hippocampus and MTL areas, you make memory worse. Okay, that was back in 2018. In the limited time that we have, I just want to zoom to the present and tell you a little bit about what we're doing now. And I'm going to just, forgive me, I'm going to zoom to here. So make sure I can see that. Yeah. So in the latest study, we had 42 patients who had LTC stimulation, lateral temporal cortex closed loop stimulation. And we found that when you stimulate in white matter tracts of lateral temporal cortex, you can produce a 28% improvement in verbal episodic memory, which corresponds to approximately 5 years of loss in patients who have an ongoing neurodegenerative process. That is, if you look at an average patient at time 1 and at time 2, 5 years later on this free recall assessment, that's what you would see over that period. That's how much of a reduction, or if you turn the clock back 5 years, that's how much better you would do. And that was statistically significant. There was no effect when you didn't stimulate near white matter, and there was importantly no effect when you did random stimulation. It turned out that beyond being near white matter, the other key variable that predicted stimulation's efficacy in improving memory was the functional connectivity between the stimulated target and the broader memory network. Okay, great. So we published a whole bunch of papers, 5 papers, 3 different data sets showing memory improvement with closed loop stimulation. But the box that you saw before is too big to put in a person's head. That big thing, it's bigger than a Sony Walkman, if anybody is old enough here to remember that technology. It's a little bit smaller than a tape recorder that we used to use when we were kids. So we needed to do something else if we wanted to deliver this therapy to older adults or patients with acquired brain injury. And so that's why I decided to try and advance this technology through the work of my company that I co-founded with Dan Rizzuto, Nia Therapeutics, and the concept is illustrated here. And I'll take a moment to actually, if anybody would be so kind as to pass this around, it's actually the device. You can take a look at if you want to open it and touch it, feel it. That's the technology that we're developing. It's an AI-guided neurostimulation system that provides a personalized therapy for memory loss. It includes a cranial implant, which is shown in A. It includes 4 thin electrodes that go into the brain, similar to the electrodes you would use in DBS for Parkinson's disease. It has a wearable earpiece, which we have now shrunk from the size that you see there into a size that's closer to a hearing aid, and a cloud-based AI platform to securely personalize the therapy. Here's a picture, so some of you are seeing it live. Some of you can see it here on the photograph. It actually was pretty hard to do this. I thought it would be easy. I was always good at math and physics and all that stuff, and I thought I could figure out how to do this. There were a lot of false starts. It took us about 5 years to build that device. We tested it in 3 sheep trials. The first sheep trial led to a major revision of the IPG. The second sheep trial worked well, but we needed to revise the EP, the earpiece. And the third trial, which is still underway, is collecting data from 2 sheep in Minneapolis. And so far, it's going very well, generating some very good data. We're not trying to improve the sheep's memory. We're just trying to establish that we can decode neural signals, that we can modulate the brain in closed loop, and that we can measure the effects of stimulation reliably. And of course, make sure that it's safe so that we can get FDA approval for a first-in-human study, which I hope we will do next year in 2026. Here is the team as it stands right now at Nia Therapeutics, which is based here in Alston, not far from where we are at the moment. And they've done all the engineering work to build the device, and I've gone on leave of absence from Penn so that I can help the company bring this into human trials and exercise it in patients first with traumatic brain injury and hopefully soon after with mild cognitive impairment and Alzheimer's disease. Thank you so much.